Okay. So here's the other. All right, so this is 6B. We're going to continue. There's a slide here to review kind of what we talked about in the first unit there. Trying to get that up. There we go. To review, remember skeletal muscle is stimulated to contract by a motor neuron. The action potential in that motor neuron stimulates the release of acetylcholine into the neuromuscular junction, the synaptic cleft there. Binding of that acetylcholine to the receptor on the sarcolemma at the motor end plate stimulates the opening of channels that allow sodium to rush into the cell, into the skeletal muscle cell. And basically what that does is stimulates another action potential on the sarcolemma at the motor end plate that then spreads out through the rest of the muscle cell along the membrane and down into T tubules, where that action potential reaches in the T tubule, basically meets up with the membrane of the sarcoplasmic reticulum and stimulates the release of calcium from there. Basically, calcium then takes that signal and binds to troponin and basically causes interactions between the thick and the thin filaments that causes the shortening of a sarcomere. We went into all the details about that in the last, the video for 6A. And in the slides for 6A. So go back and review all of that, make sure that all of these steps make sense and the details of these steps make sense, because those will be important for the exam and for the quiz for chapter 6. And there's a figure here that kind of reiterates that, right? So excitation here, this is the summary of what's called excitation contraction coupling. The coupling between exciting the muscle from the nerve to contracting the muscle by releasing calcium. So excitation, here's the motor neuron releasing acetylcholine. The action potential runs into a T tubule, stimulates calcium release from the sarcoplasmic reticulum. Calcium binds to troponin on the thin filament, moves tropomyosin away from the actin binding sites. Myosin binds to actin, pulls on it, shortening the sarcomere, right? All of that occurs to cause what's called a muscle twitch. In one muscle cell, one contraction relaxation cycle is a muscle twitch. And twitches can vary in duration, how long they last, depending on the muscle type and location, and some other environmental conditions. So basically, right here is the point of stimulus, and then contraction in like an eye muscle, super quick. So think about blinking your eye versus a deeper calf muscle that you would use to walk. So contract and relax to walk, and then contract and relax to walk. But if you need to blink your eye or move your eye to the left and to the right, really quick movement, contraction relaxation happens really quickly. So how long a muscle will contract depends on the type of muscle, where it's located, and the needs of that location. In one muscle twitch, there are 3 phases. The latent period, so the time from stimulus, from the motor neuron, right, releasing acetylcholine, to the beginning of contraction. There's about 2 milliseconds from when the nerve is stimulating the muscle to where the muscle actually starts to contract. So during that time, there's no tension developed. That's the time where the action potential is moving along the sarcolemma, muscle cell membrane, to the point where calcium actually gets released and muscle contraction can begin. So until calcium is released, contraction cannot start. So the latent period is a time from stimulus of the sarcolemma, generation of the action potential, to calcium release from the sarcoplasmic reticulum. And then contraction begins to a peak, and then relaxation is the last phase, from the peak tension to complete relaxation of the muscle cell. So here's the graph of that. So here's a stimulus. Right here would be the action potential is generated on the sarcolemma of the muscle cell, right at the motor end plate, right? The part of the muscle cell membrane that makes up half of the neuromuscular junction, the muscle side's half of the neuromuscular junction. So these 2 milliseconds from here to here is the time it takes for that action potential, the electrical signal, to move from the neuromuscular junction. What's happening? No. To move from the neuromuscular junction into the T tubules and stimulate calcium release from the sarcoplasmic reticulum, which then begins contraction all the way up to some maximum contraction, to maximum tension production, and then relaxation. And again, this time frame can change depending on the muscle. TREPI is this unique characteristic that with a second stimulus right at the end of the relaxation period. So back to this figure here. If you stimulate the muscle to contract right here, so you have one stimulus here, the next stimulus is right there, right at the end of relaxation. That repeated stimulation immediately after relaxation causes an increase in the ability of that muscle to generate force. So it increases how much tension can be produced. So here's a maximum tension for this one contraction, this one twitch. If you stimulate the muscle to contract here again, the next maximum for the next twitch will be even higher. So I'll show it to you on the coming slides. That's called TREPI. It's basically an increase in peak tension with every successive stimulus that's delivered very shortly after the complete relaxation phase is done. So in this instance, TREPI is allowing the muscle to completely relax and then stimulating it again right away. So here's what it looks like. You've got tension development here on the y-axis in the muscle, and then the time. So right here is a stimulus that causes contraction. When the muscle relaxes, you stimulate it again, and then boom, more tension development. And then you allow the muscle to relax again, but stimulate it right as soon as it's done relaxing. You let it completely relax, but then stimulate it right away. And you increase and increase and increase to some maximum amount how much force the muscle can produce in one twitch. With the second stimulus before the end of relaxation period, what happens is wave summation. So as opposed to TREPI, where you let the muscle completely relax and then immediately stimulate it to get a bigger twitch, a more forceful twitch, now we're not letting the muscle relax completely. So before it relaxes completely, you add another stimulus. And what occurs is basically summation. Think summing, like 1 + 1 is 2. 2 + 2 is 4, right? You're summing those values. Basically, the electrical signal and the contraction inside the cell is basically summing. And here's what it looks like. So the muscle hasn't had time to relax here, but then you stimulate it again, and you get a bigger force of contraction. And then you allow the muscle to relax a little bit, and then you get stimulated again, bigger force of contraction. And you allow it to relax just a little bit, but not completely, and then and you stimulate it and get an even bigger contraction. This is called wave summation, and what that does is it allows the muscle to generate more force by stimulating it more frequently. So not allowing it to relax, basically what happens here is when you're relaxing the muscle, calcium is being pumped back up into the sarcoplasmic reticulum and allowing for relaxation. Well, if you stimulate the muscle before it's allowed to completely relax, some of that calcium stays behind in the sarcoplasm and can contribute to the next contraction. And what that does is a little bit of calcium is left here that the muscle is remaining in a state of contraction, and then you dump a bunch of calcium out of the sarcoplasmic reticulum from the next stimulus, and you get a greater stimulus. So basically, a greater response. So basically, calcium is a contributing factor here. The more you leave behind from the previous contraction, the greater the next contraction will be. Incomplete tetanus. So this is even if we're going in step-by-step process to get to the point where we're going to talk about tetanus. So even more rapid cycles of contraction and relaxation produce almost peak tension, but there's still some level of relaxation. That's called incomplete tetanus. So what's happening is you've got stimulation, stimulation, stimulation. Here's wave summation, wave summation, wave summation, and then all of a sudden you reach this peak tension and you're stimulating the muscle, allowing it to relax just a little bit, but stimulating it so much that it doesn't have much time to relax, and you get this peak tension. This is incomplete tetanus. Think about what tetanus is and why you get a tetanus shot. Tetanus is complete contraction of a muscle with no relaxation. So incomplete tetanus is a little bit of relaxation, but not much. You get this big, huge increase in force of tension force, in incomplete tetanus. And then complete tetanus results in constant and continuous contraction with absolutely no relaxation. So no calcium ions are being taken back into the sarcoplasmic reticulum. They're all just flooding the sarcoplasm. And you just get one huge contraction with a really high frequency of stimulus. There is a toxin, I believe it's a virus, that causes, that basically this is why you get a tetanus shot. If you don't get a tetanus shot and you you contract tetanus, you can basically cause a hyperstimulation and an overcontraction, a contraction of your skeletal muscles to the point where you can't relax. One of the first symptoms of tetanus poisoning is tightness in your jaw. So the muscles that allow you to chew and talk start to get tight, and you eventually can't open your jaw. So Google tetanus and look that up and look at how severe the consequences can be. Skeletal muscle basically is composed of thousands of muscle fibers. We talked about, up until now, one muscle fiber and one twitch, one contraction relaxation cycle, and how one neuromuscular junction can stimulate, basically one motor neuron can stimulate a muscle cell to contract. And every single muscle cell has one neuromuscular junction. So one motor neuron is innervating, one branch of one motor neuron is innervating one skeletal muscle cell or fiber. But a motor neuron can branch at its very terminal end. So we talked about axon terminal. That axon terminal, like the axon of a neuron, is a long extension from the cell body. That axon can branch, and when it branches, the branches can all basically create a neuromuscular junction with a bunch of different muscle fibers. And one single motor neuron and all the muscle fibers that one motor neuron innervates is called a motor unit. And one motor neuron can innervate just a couple of muscle fibers, or one motor neuron can innervate thousands of muscle fibers, or hundreds of muscle fibers. So the size of a motor unit can basically can be different from muscle to muscle, and it's basically indicating how precise the movement is. So you have really small motor units where you need more precision, like in your fingertips when you need to pick things up, or in the muscles that move your eye. They have really small motor units, meaning one motor neuron innervates only a couple of muscle cells or muscle fibers. But in like your quad muscles, the muscles in your thigh, they're huge muscles, and they need a lot of force production and just a lot of gross movement, not fine precision. Those motor units are going to be huge. So one motor neuron will innervate 10s, 20s, 30s, even hundreds of muscle fibers all at once. And smaller motor units activate first, followed by larger and then more powerful motor units to increase tension. And that increase in basically the number of motor units activated to generate force or generate tension is called recruitment. Motor unit recruitment. So take a look at this figure here. Here's one motor neuron, and then this is the axon of this motor neuron, and then it branches. And that one branch innervates one muscle cell, and another branch innervates another muscle cell, and another branch innervates another muscle cell. So all of these orange branches innervate the orange muscle cells. So that's one motor unit. The motor neuron and all the muscle cells that it innervates is a motor unit. So in this muscle, there's 3 motor units: an orange, a blue, and a purple. And motor units can be recruited. So when you lift a little bit of weight, say you lift 5 pounds, this is your bicep muscle. If you lift 5 pounds, it's not that heavy. So you may only use one of these motor units and recruit one motor unit to lift that 5 pounds. You don't need all of them to lift 5 pounds because it's not that heavy. Then if you need to lift 10 pounds, you may recruit another motor unit. So the first one will be activated, and then the second one gets activated too. So the green one is now being activated, meaning that the orange and the green fibers are all being activated to contract and lift that weight. Then if you need to lift, say, 15 or 20 or 30 pounds, you may need all of the motor units in that muscle, so all of them will activate to lift that weight. That's motor unit recruitment, the addition of more motor units to increase tension production in the muscle. Some motor units are active even when the muscle is not contracting. So you've got some muscles in your body that have motor units that are active when you're not actually contracting the muscle. And that's creating some level of resting tension called muscle tone. And it's completely subconscious. It's basically just there's some level of background muscle contraction, muscle tone, so that the motor units stay active and they maintain constant tension in the muscle, but let the individual muscle fibers relax. So what can happen is, I went ahead too soon, like muscles that of posture that keep you sitting upright or standing upright. Those muscles are always active to keep you upright, and there's always some level of activity in those muscles, but not every fiber. So muscle tone is just some level of tension in a resting muscle that allows them to kind of stay functional and stay somewhat contracted subconsciously. Contraction when tension rises, basically the muscle length changes. So think about lifting your book bag, lifting it with your bicep muscle. So put your book bag in your hand and bend your elbow and extend your elbow. So flex and extend it. When the muscle length changes, that's an isotonic contraction. The muscle shortens, but the tension on that muscle remains the same. So iso means same, tonic means tone, right? So the same tension, tonic is tone or tension. So the same amount of tension. So your book bag is, say, 30 pounds. That 30 pounds isn't going to change whether you lift it or lower it. So that isotonic muscle contraction is the muscle is changing shape. So the muscle can shorten and lengthen, but the tension on that muscle remains the same. So 30 pounds is 30 pounds. If you lift it or lower it, that amount of weight doesn't change, but the length of the muscle does. That's an isotonic contraction. So right here would be like 2 kilograms. It's about 5 pounds, 4.5 pounds. So when the muscle contracts, it lifts that weight. That 2 kilograms is still 2 kilograms. So it's an isotonic contraction, same amount of tension, but the muscle shortens. So it was longer here and shorter here, right? That's an isotonic contraction. An isometric contraction is when the muscle is contracting, but the length of the muscle stays the same. So the individual muscle fibers can shorten, but the connective tissue stretches. So if the muscle is shortening, the muscle fibers are shortening, but the connective tissue is just stretching, and the whole muscle doesn't change shape. So the tension that the muscle is producing doesn't overcome the load. So 6 kilograms here is also 6 kilograms here. The muscle is contracting, the fibers are contracting, but the whole muscle doesn't change shape. That's an isometric contraction. Think about pushing on a wall. You're not going to push that wall over, right? At least hopefully you won't. If you're pushing against the wall and you're not changing the length of the muscle, it's staying the same. Or if you're just holding your book bag in your hand with your elbow bent to 90 degrees and not flexing or extending it, that's an isometric contraction. The load isn't moving. The length of the muscle isn't changing. It's staying the same, but the muscle is contracting. Isometric contractions. Muscle hypertrophy, an example of muscle hypertrophy is this big muscly guy here. Basically repeated exhaustive stimulation to muscle, like lifting weights in a gym to the point where you're tired or fatigued, can stimulate what's called muscle hypertrophy, which is the enlargement of the whole muscle. So the individual muscle cells get bigger. They pack more contractile proteins inside them so that you can lift more weight. So you have more myofibrils. And if you have more myofibrils, that means you have more actin and myosin, the myofilaments that make up myofibrils. You increase energy stores. Here is glycogen is your energy store, which ultimately increases muscular strength, also increases the number of mitochondria in the cell. So all of these adaptations to strength training, weight training, cause hypertrophy, an increase in the size of muscles, and that's in response to strength training, weight stimulus. And where muscles can hypertrophy, they also can atrophy. And when a muscle atrophies, I don't know why that's there. We don't need to point to his wheelchair. When a muscle atrophies, it decreases the size of muscle fibers. You can lose muscle strength. Atrophy is basically a natural process of aging, but it can also happen as a result of inactivity. So if you go to the gym or if you exercise for a period of time and you hypertrophy muscles, if you stop using that, or if you stop exercising, you stop weightlifting, what's going to happen? If you don't use it, right, you lose it. So muscles will atrophy. It may be temporary, or it could be a result of injury or immobilization. Or again, if you train for a period of time and then you stop exercising, muscles will atrophy back to their original size before you had trained. So just like you can hypertrophy a muscle, it can atrophy. And when hypertrophy is the gain of more myofibrils, you can basically decrease the size of muscle fibers and lose muscle mass and tone. Some examples of problems in the neuromuscular connection, like the polio virus, attacks the motor neurons in the spinal cord in the brain that innervate skeletal muscle. And if the neurons are being attacked, those neurons can no longer communicate to skeletal muscle. And if without a neuron communicating to skeletal muscle, the muscle won't contract. So the polio virus can lead to paralyzation. Tetanus toxin, we talked about a little bit. The tetanus toxin basically causes sustained powerful contractions throughout the entire body. So basically causes a hyperstimulation of the motor neuron as opposed to a blockage of the motor neuron, where you get way too much activity from the motor neuron telling the muscle to constantly contract. Botulism, like Botox, we actually use Botox to emulate what's going on here. Botulism basically prevents acetylcholine release from the neuromuscular junction, and if acetylcholine can't be released, then an action potential, the electrical signal, can't be generated on the muscle cell membrane. Remember, acetylcholine basically binds to its receptor and then allows sodium to diffuse into the cell and generate an action potential. So without that, there's no action potential on the muscle cell membrane, which means there's no calcium release from the sarcoplasmic reticulum, which means there's no muscle contraction. Another example, so botulism is a toxin, but an actual disease is called myasthenia gravis. That results in the loss of acetylcholine receptors on the neuromuscular junction. So even if acetylcholine is being released, there's nothing for that acetylcholine to bind to. And if there's nothing for acetylcholine to bind to, then you can't generate an action potential on the muscle cell membrane, and then calcium doesn't get released inside the cell, and muscles can't contract. So a couple of different ways that problems can be caused. Polio basically shuts down the motor neuron. Tetanus hyperstimulates the neuron. Botulism prevents neurotransmitter release. And myasthenia gravis is another disease where even if neurotransmitter is released, it has nothing to bind to. There's no receptor. So a couple of diseases there that can really cause some big problems. And ATP production is required. Remember, we talked about ATP basically fuels the myosin head and tells it to swivel, stretch and recoil, and stretch and recoil. So ATP is critical for muscle contraction. And after, basically post-mortem, no ATP stops being produced. And one of the major functions of ATP is to bind to myosin, donate its energy, but then a new ATP comes in once myosin has basically swiveled and pulled on actin. That new ATP comes in, binds to myosin, and myosin detaches from actin. Without more ATP production, myosin will stay bound to actin, and what is caused by that is basically called rigor mortis. Post-mortem, after death, ATP production stops. And then the sarcoplasmic reticulum basically releases tons of calcium into the sarcoplasm, the cytoplasm of the muscle cell. So a bunch of calcium gets released. No more ATP is available, so calcium can't get pumped back up into the sarcoplasmic reticulum, and it stays in the cytoplasm, the sarcoplasm. And then that calcium causes muscles to contract. Myosin binds to actin, pulls on actin by way of the sliding filament theory, right? And then it stays contracted. Without new ATP coming in, myosin can't unbind or detach from actin, so it stays bound and you stay contracted, which is what causes basically the stiffness after death, which is rigor mortis. Okay, so that's chapter 6B. I will see you for the last lecture soon.